everyone, and welcome to today's webinar titled Accelerating Research with Fast, Flexible, and Cost-Efficient Next-Generation Sequencing on the G4 platform. This webinar is a part of the 11th annual event in the Genetics Week virtual event series. I am Kaylee Bach of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by Singular Genomics. For more information about Singular Genomics, please visit singulargenomics.com. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located on the left side of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please click on the Help Desk button located at the top of your screen within the navigation bar or from the lobby within the event. Finally, as a reminder, this presentation is educational and offers free continuing education credits. Please click on the Continuing Education Credits link located in the Abstract tab from the menu at the left of your screen and follow the process to obtain your credits. So let's get started. I'd like to now present today's speaker, Timothy Looney, Senior Director of Scientific Affairs at Singular Genomics. For a complete biography on our speaker, please click the presenter window that's located in the upper left-hand corner. Timothy, you may now begin your presentation.
Great. Well, thanks for that nice introduction, and thank you to everyone for joining. As mentioned, my name is Tim Looney. I'm the Senior Director of Scientific Affairs at Singular Genomics, and today I'm going to be talking to you about the G4 platform. So the outline of my talk is as follows. I'm going to start by introducing the G4 platform. I'm going to discuss the performance of the platform across core NGS applications. I'm then going to introduce the max-read technology for high-throughput single-cell RNA-Seq. And then finally, I'm going to conclude the presentation summarizing the learnings and introducing the pricing for the platform. So to start, Singular Genomics was founded to improve the state of the art in NGS. Since inception, we've grown to over 270 employees. We've raised $450 million to date. We're now a publicly traded company, and recently moved into 135,000 square foot office, R&D, and operation space in the San Diego area where we're located. We have a strong culture of innovation with over 170 patents issued or pending, really highlighting our emphasis on R&D and improving the science. Now, before we decided to build a new NGS platform, we talked to the community to better understand the key contributors to the long turnaround and high cost in NGS. And what we learned from those discussions was that batching related inefficiencies, but also the time for the sequencing itself were the key challenges. And so we asked ourselves, can we build a machine that delivers faster, more flexible sequencing? Because if we can do this, we could take this long and often expensive process and turn it into something that is fast and cost-effective. So doing so required innovation across multiple disciplines. We had to improve the chemistry, make it faster. We had to improve the optics, the fluidics, the image analysis, and then also the downstream bioinformatics. Well, we've done this now with the G4. This is the G4. It is the world's most powerful benchtop sequencer as a function of the speed and the throughput. It's flexible. One can load up to 4 flow cells at runtime, and each flow cell comprises 4 fluidically independent lanes to facilitate multiplexing. Apart from the flexibility imparted by the number of flow cells that one can load, we also offered flow cells of 2 types. F2 produces 150 to 250 million reads, and F3 produces between 300 and 450 million reads. Apart from the flexibility, the platform is fast. It's engineered for under 24-hour run times for all applications. That puts it at the lead of the industry across those applications. And that's made impossible by the rapid sequencing by synthesis chemistry that we developed. It's powerful. A single run can produce up to 3.2 billion reads or 480 gigabases. That's more data per day than any other benchtop sequencer. And then finally, it's accurate. 80 to 90% bases of Q30. That's made possible by a novel 4-color rapid sequencing by synthesis chemistry. The quality of the data means that the resultant FASTQ files fit seamlessly into existing bioinformatics workflows. So now a bit more on the flow cells. As I mentioned, F2 produces up to 250 million reads. So if you load 4 of those, you can achieve up to 1 billion reads per run. And you'll note that the turnaround time is under 24 hours, irrespective of the sequencing configuration, and up to 2 by 150 base pair reads are possible. Now, for F3 flow cell, as I mentioned, up to 450 million reads are possible per flow cell, and that means up to 1.8 billion reads per run. And I'll note that the quality is the same for the F3 as it is for F2, 80 to 90% bases greater than or equal to Q30. You can note on the table that the turnaround times are all under 24 hours, whether you're loading 1 or 4 flow cells. And then finally, max read. This is a unique technology that we've developed to further increase the throughput of the platform for single cell and spatial. With this approach, one can achieve 800 million reads per flow cell, or 3.2 billion reads per run. And again, the turnaround times are about 24 hours. So I want to speak a bit more about the utility in having fluidically independent lanes. Imagine that a single G4 instrument is supporting multiple research groups. Well, it's easy to give each research group a separate lane on a flow cell. Using that lane ownership model, you can be sure that the indices used by one group and their set of libraries are compatible with those of another. There's no need to worry about it because they'll be sequenced separately on fluidically independent lanes. And just to extend on that point, in some cases, you'll find that some libraries have very large inserts and others have very small inserts, such that it's difficult to multiplex them together. Well, this lane-based approach, with this lane-based approach, you ensure that that's never an issue, separate lanes for the separate library pools. And then in cases where a library requires additional sequencing depth, you can imagine spreading that library across multiple lanes. And then taking this a bit further, for the libraries that require even higher sequencing depth, one can make use of an entire flow cell for a specific library type, or even multiple flow cells. And so we like to think about the lane as a unit of NGS analysis. And to that point, as a kind of rule of thumb, F2 flow cell produces about 50 million reads per lane, and F3 produces about 100 million reads per lane. And the cost per lane is on the range of $150 to $250 per lane, while the cost per flow cell is $600 to $1,000 per flow cell. Okay. So I want to also speak a bit more about how the features of this platform play out over the course of a week. So I mentioned to you that one can load up to 4 flow cells at runtime, and each of those flow cells comprises 4 fluidically independent lanes. I also mentioned that the turnaround time is under 24 hours for all applications. So what that means is that over the course of a 5-day working week, you'll have access to up to 80 independent lanes, which means 80 independent experiments per week. And given that one can load up to 4 flow cells at runtime, it's easy to scale up or down the sequencing to fit the needs of the users. There's no cost penalty for sequencing less on one day versus another. Just to that point, it's easy to accommodate users that may wish to sequence a fixed amount every day versus users that may have a more intermittent sequencing needs. And then finally, on the slide, you'll note that I've indicated a 5 p.m. run start every day. That fixed run start is achievable owing to the under 24-hour turnaround time for the instrument. So when you're running your lab, you can ensure that if you start the machine at a certain time of the day, by that time the next day, the machine will be free and is ready to start sequencing again. So you can really organize your lab's schedule to have a specified time for sequencing per day. And so that's useful. And then one last note, another corollary of this fast turnaround time that I just want to mention is that it's really nice for applications where one is using the NGS device for technology development, because it means that you sequence your libraries on one day, you have the results the next day, and can troubleshoot, optimize a protocol before sequencing again the day after. So it's very easy in that way to achieve multiple iterations of a technology development per week. Okay. Now, apart from those benefits relating to the operations of the instrument, we've also worked hard to demonstrate compatibility with the existing NGS ecosystem. That means the library preparation providers, and we've, as I'll show you, we have excellent data showing performance across a number of library preparation kits. But it also means on the analysis side. And here we've partnered with the Broad Terra platform, AWS, and also NVIDIA, where we've made extensive use of the Parabricks platform for GPU-based bioinformatics acceleration. So a bit more on the library preparation side. We've now generated data across a wide range of key NGS applications, and some of the materials that we've produced are highlighted on this slide. It ranges from whole genome sequencing all the way down to targeted applications like somatic variant detection and microbiome. And the wealth of data that we've generated really highlights the fact that it has been easy to adapt the G4 to the existing library preparation ecosystem. And this really comes from the simplicity of the library preparation. So this slide highlights the library preparation on the G4. Starting with the image at the upper right, I have an image of the library structure of used for the G4 platform. SP1 and SP2 denote the insert sequencing primers. Paired insert reads are supported. And then S1 and S2 indicate the index read primers. Again, paired indices are supported. So SP1 and SP2 are actually the same sequence as the TrueSeq on the Illumina platform. The key difference of the G4 compared to Illumina is the flow cell binding adapter sequences. We make use of S1 and S2 sequences, whereas the Illumina equivalents would be P5 and P7. And so we've developed kits for both de novo library preparation, which is the most common approach, and I'll speak to that. But then also kits for adapting an existing NGS library to the G4 platform. So first with de novo. On the right, I have an overview of the protocol for de novo library preparation. One starts with the ligation of a stem loop adapter, which introduces SP1 and SP2 primer binding sites. That loop is cleaved to produce this Y structure, which is then subject to PCR to add indices and also the flow cell binding S1 and S2 sequences. So if you're comparing this to the Illumina library preparation protocol, you'll note that there's great similarity. Really, the key difference is that we make use of S1 and S2 adapter sequences, which are added during that PCR step, whereas the Illumina library preparation would use a different oligo, adding the P5 and P7. So it's really as simple as swapping one set of PCR primers for another. And that's why it's been really easy for us to demonstrate compatibility with a wide range of library preparation partners. Now, apart from that de novo library preparation, and I just wanted to mention that we do have kits for preparation of PCR and PCR-free workflows. Up to 96 unique dual indices are available. But getting back to the adaptation process, if one wishes to adapt an existing NGS library to the G4 platform, you have the option to preserve the existing index information. And so to do that, one makes use of a simple PCR. It's a limited number of PCR cycles, which introduces the S1 and S2 flow cell binding adapter sequences to that library. One benefit of using PCR for this process, apart from the simplicity of the workflow, is that it does a great job in preserving complexity. And one wishes to improve to conserve the complexity. That's important for applications such as somatic variant detection, where a loss in complexity could negatively impact results. And then if one wishes to rewrite the index information, we have a kit for that. It makes use of primers that bind to the SP1 and SP2 sequences. And in that manner, again, through a simple PCR process, one can rewrite the existing indices to those of your choice. So everything that I mentioned is available. These are products that are available. One can order the kits through the Singular Genomics website. Great. So now I'm going to switch to the performance of the G4 across core NGS applications. And my goal in this section is really to highlight, to convey to you that the G4 produces data that is very high quality and is quite consistent with what one would achieve from the leading NGS platform. So this table highlights the applications that I'm going to touch upon. I've set this up such that the table will indicate the approximate level of multiplexing, whether using an F2 flow cell, an F3 flow cell, or the max read approach in cases of single cell sequencing. Great, so let's start with human whole genome sequencing. The F3 flow cell was designed to produce enough reads to achieve approximately 30x coverage of a human genome. And this slide highlights that very application. Using this F3 flow cell, we sequenced a human whole genome derived from HG002 to about 30x coverage. At the bottom left, you'll note the quality scores as a function of cycle over this 2 by 150 base pair experiment. We see high quality scores greater than 80% Q30, and you can see that both read 1 and read 2 are quite high quality. In the middle, we have a plot of the observed versus predicted quality scores. We see excellent calibration of the quality scores in this platform, which is important for downstream somatic and germline variant detection. And then on the right, the normalized coverage as a function of GC content, showing good coverage of both high and low GC content areas of the genome. And then finally, in the upper right, we have a table showing the performance for SNP and indel detection. These metrics were obtained using deep variant and a custom whole genome sequencing model that we developed for the G4, which we've seen outperforms the default model with deep variant. So we see really results that are on par with the state of the art in the field. And then lastly, I'll mention that our custom deep variant model has been deployed to Parabricks, which enables rapid variant calling with a turnaround time that's as low as 30 minutes FastQ to VCF for that whole genome sequencing. Great. Now switching to exome. As we had done with whole genome sequencing, we worked to develop best-in-class methods for exome analysis. And again, using deep variant, we developed a custom deep variant model optimized for exome sequencing on the G4. And the poster thumbnail on the left highlights a presentation we gave at ASHG last year, where we introduced this new model. And as you can note on the right, the model outperforms the default deep variant model. And so whether you're doing whole genome or whole exome sequencing, you can be assured that you'll be able to achieve best-in-class results with the G4. And then lastly, I'll say that this model is also available on Parabricks, which can achieve a turnaround time of about 8 minutes FastQ to VCF for the exome analysis. Great. Now I'd like to switch to target enrichment. And this includes important applications such as somatic variant detection. And somatic variant detection was one of the top priorities for us to evaluate with the G4. And to do that, we started by creating a reference material for this somatic variant analysis. And we did this by taking 23 reference cell lines from the 1,000 Genomes Project, which have like very good annotated genomes. And we pulled those at equimolar frequency to create a material whereby there were a large number of variants at approximately 2% frequency. So that material was used for library preparation using the IDT XGen cfDNA library preparation kit, which introduces UMIs. It was then subjected to enrichment using the IDT XGen pan-cancer panel, which has an about 800 KB footprint. The enriched library was sequenced on the G4 using 2x150 base pair reads and a single F2 flow cell. It was also sequenced on the NextSeq 2000, in both cases achieving about 20,000 X coverage. For each data set, we subjected the data to a rapid UMI-based error correction using the Parabricks platform. And then finally, we called variants in each data set in exactly the same way, using vardict and really generic parameters. So the aim of this study was to say, if we take the data sets from either platform and we subject them to exactly the same bioinformatic analysis, what results do we get? How similar are the results? And this is what we see. The table on the left shows the key sequencing alignment and variant calling metrics. You'll note that the G4 and NextSeq datasets are highly concordant, both in terms of coverage, the coverage uniformity, but then also at the bottom, the precision and recall an F1 score for SNP and indel detection. Shifting to the middle, we have a scatter plot showing the observed variant allele frequencies for SNPs and indels for the G4 versus the expectation based on this pooling strategy. And we see excellent correlation of those SNPs and indels. At the right, we see a correlation of those variant allele frequencies across platform, and again, an excellent correlation in the variant allele frequencies for both SNPs and indels. So I think this is a great study because within this footprint, the panel region, there are approximately 1,800 SNPs and indels where performance could be assessed. So from this results, I think it's clear that the G4 produces results that are highly concordant with the Illumina platform without need for custom bioinformatics analysis. Apart from that, our own studies, we've also seen excellent performance with our library preparation partners. On the left, we have results from the IDT Onco Hotspot XGen Custom Hype Cap app note, where IDT saw great reproducibility and sensitivity using the G4. Shifting to the middle, Paragon Genomics, the CleanPlex OncoZoom and hereditary cancer panels detect SNVs, indels, and also CNVs. And they saw great reproducibility with the G4 and also excellent concordance G4 versus Illumina. And then finally on the right, Watchmaker Genomics, they have a DNA library preparation kit, which was evaluated, and they saw excellent concordance of copy number variants on the G4 versus the MySeq platform. So from these results, I think it's very clear that the G4 produces very accurate, reproducible results for the key oncology applications. Okay, now I'm switching to methyl-Seq. I think this is an interesting application, important application, owing to the role of methylation as a source of biomarkers for cancer and also oncology. So to evaluate the performance of the G4 for methyl-Seq, we prepared a library, an EM-Seq library from NA12878, which is also referred to as HG001. The EM-Seq kit produces data that's similar to bisulfite sequencing, but it does so using an enzymatic mediated conversion step that is gentler than traditional bisulfite sequencing. So this can generally lead to libraries that are higher complexity with a greater fraction of the material preserved. So we sequenced this library using the F2 flow cell, 2 by 150 base pair reads, and then we analyzed the data using BWA-meth and methyl-DACL. So these are very commonly used standard tools for methyl-Seq. And then finally, we compared the G4 results to those obtained from the NovaSeq 6000. So starting on the left, we have some key plots looking at the methylation context quality control. At the top, we have the percent methylated base cytosine, the percent of cytosine that are methylated as a function of the cytosine context. And as would be expected based on the biology, we see that approximately 50% of the CpG context cytosines are methylated, both from the G4 and the NovaSeq data sets. Virtually all the cytosine methylation is in the CpG context. Moving below to the PUC19 positive control, this positive control should show 100% CpG methylation, and indeed it does across both platforms. And then finally, the lambda negative control looks to be entirely unmethylated across each platform. Now, turning to the right, at the top, we have the coverage over CpGs as a function of chromatin mark. What we did is we took the annotated chromatin marks, 2 activating and 1 repressive. The K27 is an activating chromatin mark. K27 trimethylation is a repressive chromatin mark, and then K4 trimethylation is an activating chromatin mark. And we asked on all of the regions of the genome where these marks were detected, what is the CpG coverage? And we see excellent uniformity of coverage as a function of the chromatin mark, and that the profiles are nearly identical across the platforms. And so this indicates that the coverage of the genome is good, and that we see good representation of biologically relevant sequences. Now, shifting to the plots at the bottom, this shows the percent of cytosine methylation as a function of each of those chromatin marks. And I think this is interesting in that you'll note the activating marks show hypomethylation within the mark body, with hypermethylation upstream and downstream of the mark, as is expected based on the biology. By contrast, K27 trimethylation, which is associated with polycomb element, and it is thought to have a sort of antagonistic relationship with CpG methylation. Those regions show less methylation. And then the profiles are identical across the G4 and the NovaSeq. So I think this is really highlights that the platform produces good data for methyl-Seq. But you may be wondering, methyl-Seq libraries are often considered to be low complexity. Does this pose challenges for the G4 sequencer? To assess this, we performed an experiment where we titrated in phi-X into an EM-Seq library and asked how the quality scores were affected. And what you can note from this plot is that there was no impact on quality scores, whether one titrates up to 50% phi-X or uses no phi-X at all. So this indicates that one can achieve good results on the G4, even with low complexity libraries. And then if we take this, we actually decided to take this a bit further, and we prepared an extremely low diversity library. In fact, only comprising 2 unique amplicons. And we again asked how the quality scores are impacted by the phi-X spike in amount. And again, we saw that there was no impact of the phi-X on the sequencing quality. Uniformly high quality, even if one has no phi-X at all and the library is extremely low complexity. And I'll say that this was not a surprise to us based on the way in which we register clusters, but the impact is that one does not have to load a large amount of phi-X when sequencing a low complexity library. You can certainly use phi-X and it's useful in assessing accuracy, but it's not necessary. Great. Now to turn to shotgun metagenomics. This is an important application owing to the range of GC content that one can observe within the microorganisms. And the very high and very low GC content can sometimes pose problems for NGS platforms. So it's important to assess as one is evaluating the performance of a new platform. And so to do this, we prepared a library from a NIST microbiome reference material comprising 19 taxa, having a range of GC content going from very low to high. We sequenced that on the G4 and then also the NextSeq 2000. And then we asked how the representation of each of those organisms compared. At the top upper left of the slide, you'll note on the y-axis, the observed versus expected abundance of each of those organisms, while the x-axis shows the genome percentage ranging from low on the left to high GC content on the right. Each platform showed good representation. In fact, there's no statistical difference in the representation as a function of GC content. To look at that in another way, the scatter plot below shows the observed versus expected abundance for both the G4 and the NextSeq 2000. So both platforms show a good correlation with truth. The G4 a bit higher, but there's actually no statistically significant difference in the correlations between the two platforms. And then on the right, finally, looking at the correlation of the abundance of each of those 19 taxa across platforms. high correlation, indicating that the data is very comparable across platforms and there's not significant GC bias. Now, switching to RNA-Seq, I'll just briefly mention that we've seen wonderful results with RNA-Seq, both internally, but then also from our library preparation partners. So just to highlight some of those results in this slide, on the left, this shows a snapshot of results from the QuantaBio Spark RNA-Seq HMR kit and their app note. And the stacked bar plots show the relative abundance of different RNA biotypes as a function of input amount. They see high consistency across a large input range. And then in the middle here, Bio-Rad, Sequoia Complete, and Express stranded RNA-Seq kits, they saw excellent concordance of the G4 versus the NextSeq 500 for both microRNAs and long non-coding RNAs. And then on the right, QIAGEN QIAseq microRNA kit, they saw excellent concordance of the G4 versus the MySeq. And I want to mention in all of these app notes, the library prep partners made use of their existing bioinformatics analysis. There was no need for customization for the G4 platform. So you can certainly make use of common tools such as STAR and Salmon for this RNA-Seq analysis and be confident that the results will be highly concordant with what you've seen elsewhere. Great. Last but not least, I want to speak about single-cell RNA-Seq. So one of the first things we wished to do when building a new platform was to demonstrate the performance with the most common single-cell library prep kits. We worked with 10x as part of the Compatible Partner Program to demonstrate good performance of the G4 with their 3' single-cell gene expression kit. And the experiment we performed pursuant to this objective was as follows. We prepared a 3' gene expression library from approximately 7,000 human healthy donor PBMCs. That library was sequenced in replicate on the G4 with 2 separate F2 flow cells to achieve about 20,000 reads per cell. We sequenced the same library using the NextSeq 2000 and an equivalent sequencing depth. And then in all cases, we analyzed the data using Cell Ranger, followed by ScanPy and SCVI tools. So to start with the sequencing metrics, both of those G4 replicates on the F2 showed results that were in line with specification. The reads were a bit under 200 million for each of those flow cells. And then turning to the Cell Ranger metrics, we see excellent Cell Ranger QC metrics with this platform. You can note 98% valid barcodes. So, you know, just again to emphasize the quality of the FASTQ files produced by the G4. Now, to evaluate the consistency across those replicates, we performed pseudo-bulk RNA-Seq analysis. And this correlation, the scatter plot shows the high correlation across those 2 replicates on the order of 0.99, really matching expectations for such a replicate sequencing analysis. We also looked at the correlation across platforms. We saw an excellent correlation, G4 versus NextSeq 2000. And just to further emphasize the similarity of the data types, we also looked at the cell annotations across platforms. And this figure on the right shows an overlay of the UMAPs for each of those data sets with the annotated T cells, monocytes, and B cells. So we observed virtually identical abundances of these key immune cell types, indicating that the data is quite similar. And then lastly, we took this a step further and used Cell Typist to annotate each cell in the G4 and NextSeq 2000 datasets. And what we found, which is displayed in this slide, is that the cell typist annotations were virtually identical in each data set. The adjusted brand index was nearly 1, just illustrating that point. And you can note, based on the UMAPs, which are colored according to the cell annotation, but then also from the heat maps at the bottom, which show the abundance of key immune cell markers across the platforms. Now, apart from 10x single cell, we also looked at the performance of the G4 for Visium spatial sequencing. And here I have a snapshot of a study led by Ioannis Vlachos, who is one of the early adopters of the G4. And he applied the G4 and Visium to study primary sclerosing cholangitis. This is an immune-mediated disease of the liver. And what we're seeing here are tissue samples from 2 donors, P1 and P2. And we see the abundance of, on the left, scar-associated macrophages, cytotoxic T cells in the middle, and then collagen. And what we're seeing here is that the results from this platform and the Visium match the expectations based on the biology. So we knew from this these results that the G4 was performing well for spatial. And then the last thing we did was demonstrate performance with other single-cell library prep partners. On the left, I have some scatter plots from the Takara Bio Smart-Seq, both mRNA and single-cell plus, comparing the G4 to the NextSeq 500. And then on the right, this is cell typist annotations of G4 versus NovaSeq 6000 using the Parse Bio EverCode WT kit. We saw really nice results with this kit. And you can note the adjusted RAND index is quite high again. It's nearly 1. Great. So we were happy with the performance of the G4 for single cell sequencing. But we asked ourselves, can we further increase the throughput of the platform for single cell? Specifically, we challenged ourselves to increase the throughput up to 200 million reads per lane, because that would mean that one could sequence a typical single cell library in each lane of the 4 lanes of a typical flow cell. And then the other question we asked was whether we can reduce the cost down to $1 per million reads without increasing the cost of the hardware for the platform or anything else. So the solution we devised is called max reads. And max reads is an approach that allows one to multiplex at the level of a nanowell within a pattern flow cell. The protocol is as follows. One starts by loading, starts with a conventional single-cell RNA-Seq library that is divided into 3 aliquots. Each of those aliquots receives a unique set of sequencing primers through a simple PCR adaptation step. And in this slide, they're denoted M1, M2, and M3. After that PCR step, those aliquots are repooled and then loaded at very high, high seeding density onto a pattern flow cell, thereby yielding a plurality of multi-templated clusters. Those multi-templated clusters are then deciphered over 3 rounds of sequencing to yield up to 3 high-quality reads per cluster. And in that manner, the throughput of the flow cell is multiplied. And we see great results with this approach. We see very high accuracy, correlation across replicates, as indicated by the UMAP and the scatter plot at the top. And then we also see very high correlation across platforms, here showing a comparison to the NextSeq 2000. And then on the right, showing that in another way, heat maps of the key expression of key markers. So we're really excited about this new approach and how it can further improve the throughput of the platform and reduce the cost. And we're now looking at this, applying this to bulk RNA-Seq. So this is now a product. Again, $1 per million reads for single cell or spatial. It's designed for the 10x genomics, popular 10x genomics kits. 3.2 billion reads per run, or 800 million reads per flow cell. And that means that one can analyze up to 16 samples per run, assuming 4 samples per flow cell. Great. So finally, I want to wrap things up here with the conclusion section. Getting to pricing, the G4 costs $350K. That's the list price. And we have flexible financing available. As I mentioned, the cost per lane starts at $150. It's $1 per million reads for the max read approach that I mentioned. And then it's $8 approximately per gigabase. We have a risk-free acquisition. You'll only be invoiced once the machine is installed and up and running. We have a great white glove installation and a support team, 1-year warranty. And then lastly, I'll mention there are great opportunities to work together with us on sequencing projects of mutual interest. And that can be a great way to drive the science and really accelerate interesting projects that you might have in mind. And then, one other very important aspect of getting a G4 is the access to our great customer support team. They're really exceptional, and we've heard great things from our customers about our support team. So I want to mention them here. Great. So to summarize what I've told you today, the G4 delivers fast, flexible sequencing with data that fits seamlessly into existing bioinformatics workflows. You can achieve under 24-hour turnaround times, avoid the traditional batching-related delays. We see strong performance with low diversity libraries. That means you don't need to load a large amount of Phi-X. And then I mentioned the high throughput max read format, which delivers cost-effective single-cell RNA-Seq. And then lastly, I'll say that the G4 is really ideal for customers employing assays with diverse sequencing depth requirements, where you may need different sequencing throughput depending on the day. So with that, I want to thank everyone for listening, and I'd love to take questions during the question and answer portion of this presentation. Thanks, everyone.
Thank you, Timothy, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many questions as we have time for. All right, so let's get started. Timothy, my first question for you asks, what is the minimum input amount for the G4?
Great question. The library seeding concentration is from 7 picomolar to 25 picomolar for typical use cases. Most typically in the lower end, like 10 picomolar. And so I would say that's a strength of the G4 platform, and that you don't need a very high concentration library to successfully sequence.
Wonderful, thank you. I have another question for you here, and this asks, are there any unique attributes to the FASTQ files that bioinformaticians should be aware of?
Yeah, another great question. Firstly, I'll say, as you probably took from my presentation, we've aimed to create FASTQ files that fit seamlessly into the existing bioinformatics ecosystems. So at a large scale, they're quite similar to what you may be familiar with from the Illumina platform. I will say, though, that we have observed modest improvement in deep variant through the implementation of a custom deep variant model that we produced in-house. Just to expand on this a bit more in terms of the error modes on our platform, the sequencing error modes, like Illumina, the most common error mode is a substitution sequencing error, where insertion deletion errors are comparatively rare. And I think that's the similarity in the error modes is one reason why we can take off-the-shelf germline and somatic variant detection software and use it successfully with the G4 without need for modification.
Wonderful, thank you. Another question for you here. How is the performance of the G4 for ChIP-seq?
Yep, great question here. We see excellent performance in terms of the data recapitulating the expected biology, but then also cross-platform concordance, G4 versus Illumina. I've heard from customers that they're seeing great performance for all sorts of ChIP-seq, including modern, the most cutting-edge assays like cut and run. So I think you can have good confidence that you'll have nice ChIP-seq data with this platform.
Wonderful. Well, it looks like that's all the time we have for today. So we're going to go ahead and wrap up here. But Timothy, do you have any final comments for our audience?
Yeah, I'll say that I presented quite a bit of different data types during this webinar. And I'll just say that we have a great deal of the data available at the Singular Genomics website. You can download the data, explore it yourself. We also have Jupyter notebooks describing all of the analysis steps that we took to produce the figures I shared. So I think that's a nice way to explore the data yourself and get familiar with the G4.
Wonderful. Well, thank you again, Timothy, for your time today and your important research. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. I'd also like to thank our sponsor, Singular Genomics, for sponsoring today's webinar. Questions we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 2 years until May 18th, 2025. Labyrinth will alert you via email when it's available for replay, and we encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.